Today we are going to discuss basic properties of T4 bacteriophage, which is also known as Escherichia virus T4. The host for this virus is Escherichia coli. That's why it is also known as Escherichia virus T4. So this virus or this bacteriophage T4 is not infectious for human, but it is infectious for Escherichia coli. So the target organism for this virus is E. coli, or the host for this virus is E. coli. Bacteriophages or phages were generally isolated from the natural resources such as human sewage, and they are studied early 20th century. Definition of the bacteriophages is: these are those viruses that infect bacteria. Here the suffix phage comes from the Greek of eating because of their ability to eat or lyse bacteria. T4 bacteriophage is one of the 7 Escherichia coli phages. So there are T1, T2, T3, T4, T5, T6 and T7 phages. So here T stand for the type. So T2, T4 and T6, these are called the even bacteriophages. So here the T4 is one of the 7 bacteriophage. It is considered as an even bacteriophage. So these T2, T4 and T6, which are the even bacteriophages, they are similar in structure, antigenic response and genetics. So these three are quite similar with each other. Bacteriophages T4 is classified as a member in the Myoviridae family of the Cordovirales order because it has contractile tail. So this thing we will discuss in detail in the next slide. So basic structural properties of T4 bacteriophage. So this is the diagram of T4 bacteriophage, simplest diagram. So here we will discuss its structural properties one by one. First is the biennial symmetry. This T4 phage has a biennial symmetry because it has a head which is like an icosahedron. So here we can see this particular structure. This is head and this resembles an icosahedral. So it has a icosahedral shape head and it has a tail. So we can see this particular structure. This is the tail. So this particular region is the tail and this tail is helical. So basically it has head and tail. The icosahedral head contains double-stranded linear DNA as a genome. So we can see this is the DNA molecule which is the double-stranded and linear, and this DNA remain in the head of the bacteriophage. Tail is composed of a collar. So we can see a collar is here at the starting of the tail, and this collar joins head with the remaining part of the tail. So this tail here you can see this is a hollow tube like structure and it has a sheath surrounding to it. So particularly these rings you can see, these are the sheath which are made up of particular type of protein and they surround the hollow central tail. So this tail is hollow so that the phage can inject their genetic material into the host through this hollow tubes. At the base of the tail, here we can see a base plate. So this particular structure is a base plate and this base plate is hexagonal in the case of T4 bacteriophage. The sheath of the tail is made up of 144 copies of a particular type of protein. The name of protein is GP18, and these 144 copies are arranged in 24 rings. So we can see so many rings like a structure. The number of these rings is 24, and each ring contains 6 copies. So in this manner, total number of copies of this GP18 protein in the tail are 144. In T-event phages, the base plate is hexagonal and has a pin and joint tail fiber at each corner. So here, if we consider this as a corner, so this is a pin and this is a tail fiber. So a huge long tail fiber is there at the corner of the base plate. So total number of tail fibers are 6. They are arising from each corner of the hexagonal base plate. The tail fibers has a particular function. They are responsible for the attachment of virus to a proper site on their bacterial host. So these tail fiber recognize their host and they attach on their host so that they can replicate inside the host. They can proceed their life cycle. So T4 is a relatively large virus particle, and if we talk about the dimension of this virus particle, it has 90 nanometer wide and 200 nanometer long structure. The mature phage has a head which is an icosahedral structure, and the dimension of this head is 1150 angstrom long and the width is 850 angstrom. And inside this head, the genetic material, which is the DNA, is there. The DNA of this virus is 168 kilobase pair long, and it is a double-stranded structure. This DNA contains 289 open reading frames. It means that this genome can encode 289 different types of proteins. So if we talk about the dimension of the tail, it has a contractile hollow tail and it is 925 angstrom long and 240 angstrom in diameter. So this tail is contractile. This tail can reduce or it can increase its size depending on the presence or absence of the host. The dimension of hexagonal base plate is 270 angstrom high and 520 angstrom in diameter. So as we discussed earlier, the hexagonal base plate bears 6 long tail fibers. So these are the tail fibers. 6 tail fibers are there, arising from each corner of the base plate. So the dimension of these tail fiber, or the length of these tail fibers, are 1450 angstrom. The important property of T4 phage is T4 is capable of undergoing only a lytic life cycle and not a lysogenic life cycle. So this is one of the distinguished property of the virus, that it can follow only the lytic life cycle.